All right, thank you, everybody. My name is John Birchall. I'm the marketing manager here at JASCO. Today we've got it's the first of our webinars this year, and it's part of a series that we'll be doing throughout the year. And in this particular webinar, we've got our two application specialists in vibrational and electronic spectroscopy, and they're going to be introducing both the use of IR spectroscopy and circular dicroism for protein structural analysis. If you've not been to one of our webinars before, you can find them later on online. They're accessible through our website and also through our YouTube channel. And if you go to both of those, you can find more information and more more recorded webinars and application notes on the same topics. Throughout this, there will be a Q&A, an opportunity to answer questions, and we will answer those at the end of the presentation. So feel free to ask questions as we go along, and we'll choose a selection of those at the end that we can discuss with you. So without further ado, I'd like to introduce Dr. Carlos Mourillo, who's our vibrational application spectroscopist, and he'll be talking about the use of IR for protein structural analysis. Thank you, Carlos.
Hi, hello, everyone. Thank you. Good afternoon. Thank you, John, for that introduction. So today we are going to be talking about the protein analysis using a couple of spectroscopy techniques. The agenda for today is going to be we are going to be talking about proteins, what is important to study proteins, also secondary structure estimation using FTIR and using circular dicroism. So for starting, what are proteins? Proteins are just large, complex molecules that play many critical roles in the body, and they do most of the work in cells that are required based on their structure, function, and all the regulations for our body tissues and organs. Functions of proteins, you can see them in the slide. There are several protein functions, but for example, I'm just going to mention the first one. Is like the catalysis, and one of the functions of the proteins is to perform this catalysis in the human body as well. You can find the same in the animal and plants, and a protein that perform these catalysis are called enzymes, and the enzymes are going to facilitate and accelerate these complex chemical reactions that they are required for growth and body maintenance, such as synthesis, oxidation, and reduction inside the body. The sequence of proteins. Proteins are made of units or small units that they are called amino acids, and these amino acids they are connected in what we call amino acid chains, and the protein is very important. What is the sequence of these amino acids, and actually they are going to govern what is the function of the proteins. There are currently 20, or there are studied 20 different amino acids that can be combined to make this protein, and the sequence is going to determine what is that protein unique three-dimensional structure, and therefore their function. So what are the protein structures? There are 4 different types of protein structure. We have the primary, and this primary is basically what we know as what I mentioned before, the change of the amino acid. Also, we have a secondary structure, and in this secondary structure, we found like two main structures. One is called alpha helix, that we see this structure here is alpha helixes. We have the beta sheets, that they are these platelets that you can see in yellow in the slide. We also know beta turn or random, that is I'm labeled here as an other. That is for secondary structure. The tertiary structure is just the interaction between these two main motif that I mentioned before, and the quaternary structure is just like the arrangement of these subunits and in the protein. So what are the different areas to study proteins? First, we have the pharmaceutical area or field that we have with the development of new medicines. Also, we have the food industry that they're usually mainly with the design of new foods, and it basically they use it due to the structure of the protein, and also it provides specific functions in the water and fat binding, so it can emulsify and create foams or gelling for specific foods. We have the hospital and clinic and laboratories that mainly work with genes and gene-related disease. Besides that, of course, we have academia in the field of biochemistry or biophysics, that they work with the determination of the structure and the function. And finally, we have different other applications, and more recently in the sensor area, which the proteins can be part of a sensor or can be a stand alone. So structural study of proteins, the primary structure that I mentioned was the study of these sequence of amino acids. One of the most common technique is the Edman method. We have the secondary structure to study what I mentioned before, the motif like is the alpha helix, beta sheet, beta turn, and other. We have 4 different techniques. We are going to be talking about FTIR and CD. For the tertiary structure, we have other techniques like X-ray, NMR, and computer simulation. And finally, for quaternary structure, the most common is NMR and also computer simulation. So in the protein secondary structure estimation, I'm going to compare the techniques and then talk about the secondary structure estimation program using the technique of infrared, and then making a comparison between all the techniques at the end of the webinar. So I'm going to be talking mainly about FTIR and comparing to X-ray. X-ray is a technique that is very well known. Basically, we are going to be looking at solid samples that they are crystal, of course, because we are talking about solid samples. The concentration is 100%. In X-ray, it's possible to have a temperature control, also do mapping. It's a standard method, and the information that we can get from proteins are secondary structure, tertiary structure, but it's a quite expensive technique compared to IR. In the case of IR, we can measure solid samples as well as in X-ray, but we can dilute these samples, and because they are diluted, we have the possibility to study amorphous samples. Also, the concentration can go up to 5%. That is very common, but I'm going to show that we can go even lower than that. Of course, it is possible to do temperature control in the case of if the user has an FTIR microscope, it is possible also to do the mapping of the samples. Secondary structure, yes, under study, and this is what I'm going to show how we estimate this secondary structure, and it's relatively inexpensive technique compared to X-ray. So this is IR spectrum of proteins. This is a standard spectrum, IR spectrum. In the y-axis, we have absorbance. In the x-axis, we have wave numbers. For those that they are not familiar with the IR technique, I invite you, as John mentioned, to come to our website and look and watch different webinars about the theory and instrumentation of IR. But in this, for this webinar, what we use to study the proteins is the amide-1, and this amide-1 is the peak that it goes from 1700 to 1600, and this is a carbon double bond O peak, and actually in this peak is contained all the information of the secondary structure, especially alpha helixes, beta sheet, beta turn, and other. And the techniques that or the algorithms that they are used for the estimation of the secondary structure are the principal component regression that I'm going to be mentioning as a PCR, and the partial least square, that is the PLS method. So this is the interface of our software, and in here we can select the data. Of course, this is the spectrum that you want to be studied. The two algorithms can be chosen here, and the method is just it's a drop down. We can subtract the buffer. Usually the proteins are dissolved in a buffer, and we can subtract the water vapor, and we can do other corrections. We have the results here. The left-hand side, in the right-hand side, we have the measurement of the spectrum that I mentioned before, 1700 to 1600. Calculated spectrum, this is the deconvoluted spectrum. So we have here the peak that correspond to alpha helixes, beta sheet, beta turn, and other. But we have the error that it was used for the calculation of that estimation. So how do we do that? Since this technique is based on Beer law, and the Beer law can think of a line, the technique that we use is just we use a matrix, and this matrix is conformed by 17, the information of 17 different proteins, and this data comes from the X-ray standard data. The IR data of these 17 different proteins is measured, and with these two data, so it's the coefficient matrix is calculated. So the next things to do is, since we have the information of these coefficients of these matrix, the user next measured the infrared data of the unknown sample, and from here, what is calculated is the secondary structure. So this F1 here would be alpha helix, beta sheet, beta turn, and random, and this is only one, the information from only one protein. So one example of this result, if we look at the lysozyme is a very well-known protein, very well studied. Here there is a comparison between the X-ray data and the infrared data using the algorithm of PCR. In blue, we have the X-ray. In yellow, we have the infrared. If we can see in the first graph for alpha helix, we have a very close result, where in the X-ray, we have something like 30% of lysozyme is 30% conformed by alpha helixes, and the result of infrared is close, is something like 39. So since this is estimation, the data in X-ray is a solid data, and the data that it was collected here is using transmission that is a diluted sample. It's a liquid sample. You are going to find some discrepancies, but what is important is that we can look in the tendency of these two results. For example, the lysozyme is the trend is going to be mainly enriched with alpha helix. Besides that, I think that the secondary structure estimation can give an idea how a protein is going to fold when it's under thermal load. Here we have more information about other proteins, and the results are similar. We can look into what is the trend on each measurement. So for those measurements that they are really low in concentration, JASCO, we offer a high sensitivity ATR for liquid samples, and we are going to combine these with FTIR bench and the ATR Pro Penta, that is this accessory that we see here, in combination with an MCT detector that is an affordable type, where it is a high sensitivity detector. So the Pro Penta has a pentagon shape, and it has 14 different bounces. So it's a multi-bounce ATR crystal that is going to increase the signal. And here you can see where are the points where the IR signal is in contact with the sample. Of course, these, due to the size, is going to enable the possibility to run samples with a very, very small, and because it's combined with a high sensitivity detector and a multi-bounce, we can run samples that they have a very low concentration. For example, here we have an example with a lysozyme that is 0.01%. We can see how the spectrum of lysozyme and water overlaps, but when they are subtracted, we can see the difference between amide 1 and amide 2. And when we do the calculation of the using the secondary structure estimation software, we can see that the lysozyme is the peak is deconvoluted, and we have the results, and the result that we have is that the ratio, or the percent, is 40% for alpha helix. That is really close to what it was measured before. And actually, alpha helix is lysozyme is a protein that is rich on alpha helix. With this is the part related to IR. I'm going to pass the presentation now to Dr. Forrest.
All right. Yeah, thank you, Carlos, for that really excellent description of sort of protein structure, and then how IR is used to determine the secondary structure of protein. So in my section, I'll be going over, as Carlos discussed, a secondary structure estimation through a circular dichroism, or a CD standpoint. And the way I'm going to be going through that is I'm going to give a really brief overview of protein structure, mainly as it pertains to CD, since Carlos has really given this excellent description of how proteins are ordered. And then after that, I'll get into how secondary structure estimation with CD works. And what we'll find there is actually it's really analogous to what's done with the FTIR. which is one of sort of the strengths of having this in-house algorithm that we have. And then I'm going to give a sort of concrete example of use of that secondary structure estimation software in a high throughput setting where we're looking at a high number of samples that we want to get secondary structure estimations on very quickly. So first, I'll go over sort of the brief protein structure, just to lead in how we actually look at that with CD. So like Carlos mentioned, we have this primary structure. What we're going to be investigating in circular dichroism is sort of one of three different things. There's the secondary structure, which is that alpha helix beta sheet content Carlos mentioned earlier. With CD, you can also look at tertiary structure, or how that protein is folding on a larger scale. And then you can also look at quaternary structure. And one of the nice things about CD spectroscopy as well is that it's really well suited to looking at changes in environment. So altering things like salt concentration, pH, and how that affects the protein structure are really easy to look at with circular dichroism, which I'll also briefly touch on before moving into how we perform secondary structure estimation through CD spectra. So I mentioned these three regimes that we can look at with CD measurements. Most important to this talk is the far UV region, which is anything less than 260 nanometers. This is really where we get a lot of our information on alpha helix content, beta sheet content, if there's random coil present or beta turns. In addition to the secondary structure, we can also look into the near UV to get an idea of what the tertiary structure is like. So this is looking at the amino acids that are aromatic, so phenylalanines, tyrosines, and tryptophan. And this really tells us how those aromatic side chains are being affected by changes to environment. So things like unfolding due to pH, you can look at this tertiary region. And then there's also this near UV to visible region on the right-hand side here. And this is looking often at prosthetic groups, so things like hemoglobin that have iron bound, and really anything with an active site with a metal bound ion. Often this visible to near UV region will give signal that'll give some idea about how that structure is being altered by whatever that prosthetic group is, which is often an ion. But here we're really going to hone in on the secondary structure and how we can look at that with circular dichroism. So first, I want to go into a sort of electronic transitions because that's really what we're looking at here. So if you think about a UV visible experiment, you have a ground state, this S0. You have a higher energy state, this S1. You pass light through your sample, and excitation takes place, and then you simply get an absorption spectrum, which is analogous to or related to the energy of the photons exciting your sample. And you can relate this to the concentration of your sample with Beer's law. But we're sort of missing a component here, which is that these transitions don't just take place because the energy gap is a certain energy value. You have to have overlap of a transition dipole moment. So if you think about your molecule, it has an electron cloud around it. Here we have a very simple sort of atom picture. And when we introduce this electric field or our light source, that's going to distort our electron cloud. And this change in the distribution of electrons is what's known as the transition dipole moment. And we can assign that as a vector. So it's a vector quality with direction. And so now it becomes very important to know not only what energy wavelength are we working with our light source, but the direction of that electric field from the light also is going to play an important role, specifically for dichroism studies. So if we think about polarized light, for our UV visible absorption spectrum that I just showed on the previous page, we're really working with an unpolarized light source. So we don't care how the light is polarized in this case. We only really are interested in how much is being absorbed. But we can start to polarize light. So linear polarization, here the energy or the electric field vector is oscillating on the y direction. This is linear polarization, and this can be used to study linear dichroism, which is a fairly niche method. But circular dichroism, instead of looking at this linear polarization, is instead going to look at this circular polarization here. So you can see this dashed line here is the direction of our light vector, and we can see that it sort of corkscrews around the z-axis, almost like if you think about pulling a sunky at two ends. That's sort of the direction that this electric field is taking when it's circularly polarized. And circular dichroism measurements are going to focus on the absorption of circularly polarized light. So the thing we're really looking at with circular polarization absorption here is we have right-handed circularly polarized light, which I'm showing here in blue, and that's going to be moving clockwise in this case. And then we'll also have a corresponding left-handed circularly polarized light, which will be moving counterclockwise. And if we have a chiral molecule, like this amino acid residue here, there's going to be preferential absorption of one of these polarizations. In this case, our right-handed circularly polarized light is preferentially absorbed. And we can actually go back and relate that to Beer's law. So now we have two absorption sort of values for this left-handed and right-handed. And we can get, instead of just an epsilon value, we can get this delta epsilon. And now we really have a quantity that'll help us determine structurally what's happening here. Because these chiral molecules, their structure is going to influence how much of this absorption is taking place and at what wavelength that absorption is taking place. So in terms of proteins, we can think of very simple cases, which I'm going to give here. So on the left-handed side here, we have hemoglobin, which is an alpha helix rich. In the middle, we have IgG, which is a beta sheet rich protein. And then all the way on the right, we have a trypsin inhibitor, which is more or less disordered. So it's a random coil. And what we can see is with the trypsin inhibitor, we really only have one key feature, which is this 200 nanometer absorption. And because CD is a different spectra, not only do we have peak location to work with, we also have direction. So for this random coil, we have a negative going peak around 195 nanometers. But if we look at beta sheet rich proteins now, so this IgG, we have a feature at the same region as this random coil, but instead of being a negative going feature, it's now a positive going feature. So there's clear differentiation already between this beta sheet and this random coil that helps us determine which we have. In addition, we also have this beta sheet has a negative going peak at 217, so we're seeing a new feature. And then for alpha helix, we can see that there's really no clear peak location at 200 nanometers like there was for the beta sheet or the random coil. And now instead we get this positive going band at 192, and we actually have two negative going features at 222 and around 208 nanometers. So here we can already start to see how secondary structures are going to give really nicely resolved and differentiable CD spectra. So if we look at a CD spectrum, we can already sort of get an idea of, well, this is an alpha helix rich protein because I'm seeing these two bands, or maybe we have a random coil or disordered protein because we're really only seeing that negative feature. And because these features are so easily differentiated, we can start to change a lot of parameters in our CD experiments to get an idea of how things will change the secondary structure. So CD, it's really easy to measure dynamic processes, things like folding, ligand binding, stability with temperature change. Here, this is a folding experiment using stopped flow. So we can measure very fast processes of CD. Here in this example, we have a two-step folding that's taking place, and we can very easily see that with circular dichroism. Additionally, we can also do temperature control or titration experiments. So here we have a initially random coiled protein, but as we add high amounts of acid, we actually start to force this into an alpha helical conformation, and these spectra are really nicely resolved and easy to show that transition taking place. And then the other nice thing about CD in comparison to other methods is you have a wide variety of path length selection, which allows for a lot of flexibility in concentrations. So we can use very short path lengths to get up to 2% protein, so bringing us closer to where the IR region was with what Carlos was showing. But we can also use very low concentrations as well by using higher path lengths, which is really nice for circular dichroism measurements. So with that sort of overview of circular dichroism from a secondary structure standpoint in general, I want to get into the specifics of how the estimation is handled, which is actually going to be fairly straightforward and simple because it's essentially identical to the process that Carlos has already outlined. So in the case of the CD multivariate analysis, instead of IR reference spectra, we're obviously going to be using CD reference spectra. So we have a data set of 26 known compounds. We know the secondary structure percentages from X-ray. And we can use this conjunction of the CD spectra with the percentage of X-ray secondary structures in order to get that coefficient matrix that Carlos was previously describing. Another nice thing about this method that I want to touch on is that for both of these softwares, they start with a sort of generic reference database, but the calibration models can be altered to include further CD spectra or IR spectra if you supply them with the secondary structures of those known compounds. So once that secondary structure model or coefficient matrix is developed, all we have to do is take our CD data, plug that CD spectrum in, and then it'll automatically give us our percentage of content. So I've shown here, basically the whole process is very identical to the way the FTIR algorithm is done. So once you know how to use one of these software packages, basically it's identical, the process that you go through. The only thing that's different is the sample measurement. So with that description of the secondary structure estimation software, I want to give an example of how this can be effectively used in CD. So a sort of large market right now in terms of what people want is they want very high throughput. So if we can do things faster and more efficiently, that's going to be better overall for measurement purposes. So with high throughput systems, we can measure a large number of CD samples. And not only is the sample measurement automated here, we're also going to automate the analysis as well. So we're not going to have to go through and do a lot of sort of number crunching before we can get our secondary structure estimations. The way this is going to work is that in our CD estimation software, or the high throughput systems, we can supply a path length here. So in this case, we're going to be using 1 millimeter path length. And then we can also start to give it our mean residue molar concentrations for each sample before the analysis takes place. And what the secondary structure estimation software is going to do is it's going to take this path length and this molar concentration. It's going to input those in order to get our ellipticity spectra, and then it's automatically going to calculate the secondary structure percentages from those values that we've given it. So in this case, we're going to run 8 different samples collected in fast succession with this HTCD setup. And then the secondary structure estimation, like I said, is going to be carried out automatically. So there's no need to sort of take your spectra, put it in the program, and then run things. It's automatically going to carry that out. And what we see is very similar to what Carlos showed previously. The measurements match up really nicely to the X-ray. And overall, the matching is quantitatively the same. So if we have beta-rich structures, we'll see their high percentages in beta sheet. And if they're alpha helical, we're going to see that there's a really nice match in the alpha helix content. So with that overview of both methods, I also want to conclude by giving sort of a wrap-up of the contrasts between these two methods. First and foremost, deciding between FTIR and CD, concentration plays a big role. So CD concentrations are limited to this sort of below 4% region, which for most protein work is fairly good. Typically, you don't need higher concentrations than that unless you're working with something that's really concentrated for whatever purpose that is, or if you're working with solids, in which case FTIR becomes a more obvious choice. And so with both of these methods, we can work with liquids or solids. In the case of CD, solids can be done with diffuse reflectance, but that becomes a little bit more tricky. So FTIR is usually better signal to noise wise with a solid sample using an ATR like the Penta that Carlos showed previously. Both methods are capable of doing temperature control, but CD has the capability to do these high throughput measurements and auto titration. In contrast, the FTIR is capable of doing these mapping experiments. We can actually map out secondary structure as a function of location on your sample. The secondary structure database statuses for both. So the FTIR databases are under study, but as we sort of demonstrated here, it's becoming extremely effective and is sort of a nice orthogonal analysis method in comparison to traditional CD. And then on the CD side, one of the things to note here is that there's sort of multiple options for analysis. So I've highlighted here our JASCO multivariate software, but in addition to this, there's also BestCell, which is sort of intended for beta-rich heavy proteins. It's an online algorithm that's free to use. And so algorithms like this can be used in conjunction with the JASCO multivariate to sort of double check results and make sure estimations are sort of correct in what you believe that they should be. And then, of course, with the information, CD can give us that secondary structure and tertiary structure, and FTR can sort of get its secondary structure estimation and other types of information as well. And then, of course, FTR on the whole is a good deal more inexpensive than CD measurements, where CD sort of leans on the higher $ end of things, just because of sort of the variability in measurement types you can do with CD. And so with that wrap up of secondary structure estimation from an FTR standpoint and a CD standpoint, me and Carlos would be happy to sort of go through and answer any questions you have about either of these methods or how they could possibly work in conjunction. But thank you for attending.